The fundamental principles and fundamental biological and biochemical operations are often ignored with the advance in technology. And that's a tragedy. And I have interest in bioinorganic chemistry as a chemical standpoint of view, with the responses in biological systems and the reactivities in biological systems and the responses of biological systems to the trace metals. Then later on, I switched to lipid biochemistry because I was interested in membranes, and I went to Hormel. Hormel Institute is the only lipid institute in the world, so to say. We brag like that. And this was funded by the Hormel company, and later on, University of Minnesota took it over about 60 years ago in the southern part of Minnesota. And I was really fortunate to learn the basics of lipidology there with the celebrities in lipid chemistry. And then later moved on to the cellular systems at Johns Hopkins University, looking at the lipid signaling. Signaling, actually, could I have the slides, please? Usually these are intricate networks in the biological systems. Actually, this started with enzymology. With enzyme kinetics, actually it is so difficult to measure kinetics in intact cells and intact tissues. But usually what the enzymologists do is they actually isolate them and study them either in the isolated enzymes or isolated cells or isolated tissues. That's what the greatest discovery was by Sir Hans Krebs, who actually discovered Krebs cycle. He didn't do rocket science. He just took the pigeon breast muscle, used a Warburg apparatus, simple Warburg apparatus, used a Mercury manometer, and just measured the oxygen consumption. And that's how he discovered adding substrates to the breast muscle tissue. An elegant work was done by Krebs, and now that is the TCA cycle. And then the enzymologists came, isolated these enzymes, and then looked in the test tube. What are the concentrations required for the substrates? What are the products? What is the saturation kinetics? What is the kinetics? What is KD? What is KM? How these substrates regulate? What are feedback inhibitions? What are activators? What are inhibitors? What are active sites? What are prosthetic groups? What are coenzymes? And what are the regulators at the genetic level? And the genetic material discovery by Watson and Crick revolutionized much more. Then later on, all the genetic engineering techniques have revolutionized. One thing is in the metabolism sequence. Another, credit to be given is to Melvin Kelvin at California. He was the first one who used radio tracers. Hats off to that gentleman. And those radio tracers revolutionized the entire field of biochemistry. Not only biochemistry, the entire field of biology. Simple dot reaction. He used C14. And then the whole area of metabolism exploded with the discovery, of the dot cycle and with the discovery of the dot cycle and the introduction of C14. So what is the essence of this small historical account? Until 20 years ago, people were looking at feedback regulations and the enzymes and all these kinetics and how these are regulated, gene action, gene interaction, and expression of genes and all those things. Suddenly, a group of bright people came. These are nothing but signals. Cells have signals. So there are upstream signals, downstream signals. These regulate. They don't constantly fire. There are switches. These switches regulate these signal transductions. One of the most important elements in the cell is the cell membrane. A lot of people think that cell membrane is nothing but a barrier. It's a boundary. Cell membrane we think as a phospholipid bilayer with integrated proteins and other biochemical components in them. There are receptors on the cells, and these receptors are important. They bind to the effectors, and these effectors, when they bind the receptor complex with the substance, will trigger the reactions. And in addition to that, there are transporters. These cell membranes have transporters, and these transporters allow materials to go from outside to inside and inside to outside, efflux and influx. These are maintained by the transporters. And not only that, cell membrane has 30% cholesterol. Caution, people who take statins. I'm not a clinician, I'm not a cardiologist, but something always bothers me, that people who take statins, who are given statins, they're inhibiting HMG-CoA reductase, which is the rate-limiting enzyme throughout the body, which synthesizes cholesterol from the precursor. Why do you want to unnecessarily block HMG-CoA reductase throughout the body to lower blood level of cholesterol? Coming to the transition metals and toxicities, people always measure blood levels of transition metals. Blood levels do not reflect the tissue levels. I'm sorry, I may be wrong, please correct me. And you do not want to come to an inference or a conclusion by looking at blood levels, plasma levels of these transition metals. Not only that, even nutrients, because it's not steady state equilibrium. Tissues incorporate them, tissues are loaded with these, but the blood is different. Okay, that's what happens. The blood cholesterol level is 100. Who set this value 100? You are 100, I am 150, she is 190. We have biochemical individuality. Roger Williams coined the term 30 years ago. Every individual is different. Now they're talking about personalized medicine. That means every person has a tailored requirement for medical or clinical treatment or diagnosis. So when you talk about personalized medicine, a tailored treatment, a tailored diagnosis that is required for each and every individual, then why don't we talk about biochemical individuality? The amount you eat is different, the amount I eat is different. What I require is different, what you require is different. But at a population level, there may be some general trends, but again, individual from individual is different. That's where the genetic plays a role. With all the explosion of molecular biology and genetics, we are ignoring the individual genotype. So basically, coming to the transition metals, again, when you look at a statistical standpoint of view, yes, there may be correlations. But again, are you going to ignore the insignificant? You're only saying, well, I don't accept the data, it's only significant, not significant. Or not significant is also important most of the times. So what happens? The signal transduction, going back again, making another leap to signal transduction, what happens in the signal transduction is very early on events are very important for a cell. If a person is exposed to mercury, it starts from that moment onwards. You don't have to wait for. This one?
Yeah, you can look there.
Can I use this one?
Yeah. This comes ahead.
Okay, thanks.
This goes ahead.
This is back?
No. Forward.
Okay.
You're back.
Oh, thanks. So this is a simplistic way of signal transduction network. And if you, yeah, it's the simplistic picture of signal transduction network. I copied it from somebody else. And the author's name is there. Now you can multiply that by at least 50. So you can't even, it's like worse than the roadmap. That entire United States roadmap, if you bring it together and you look at it for North Carolina, Charlotte at this Renaissance Hotel, that's where the PLD that I'm talking today. Some people even ignore it. Yeah, this enzyme is not important. How do you know? That's another problem. So what happens is there are personal preferences for scientists. Peter Medawar, the Nobel laureate who won the Nobel Prize for cell-mediated immunity, coined a term. He wrote a wonderful book called An Advice to a Young Scientist, which is only 34 to 40 pages long. That's it. An advice to a young scientist. In that there is a quotation, never fall in love with your hypothesis. So we have to be open-minded. We have to think, yeah, we like it, but still the other person has a different story or a different paradigm. Let us look at it. So this is what, coming back to the cells, the lipid bilayer I was talking about, these are bilayers actually, and these bilayers are composed of phospholipids mostly. There are different classes of phospholipids, and there are intercalations of cholesterol. Cholesterol is very important. 30% of the cell membrane, 30 to 40% of the cell membrane is cholesterol. In case of neurons, very rich. Neurons don't make cholesterol. Astrocytes make them and pump them into the neurons, and the neurons have incorporated the cholesterol from astrocytes. And now, interestingly, the sleep-wake cycles, the sleep patterns are regulated by neuronal cholesterol. And that's the latest discovery now. So again, think back of how do you regulate your cholesterol levels with statin, and that will alter the neuronal cholesterol. And now it is becoming more evident that if you take statins, myalgia is a result of statins. So be careful. And that's what, just a caution I give. So what happens, this cell membrane I coined, this is the gateway of the cell. Anything that impinges, that goes into the cell sees it, that happens right here. This is the gateway. It has to happen at the door. So that important it is. And there are other integral proteins and all those things. The receptors are here. There are glycolipid complexes here. There are glycoconjugates here that actually bind to different substances and how the cell maintains. So one of the interesting things is, since I told you that it has a phospholipid backbone with different phospholipids in it, there are several enzymes that are operating the membrane dynamics. What happens, membrane is on a constant flux. It has to degrade, it has to metabolize, it has to reform. Very quickly, it actually reshuffles, flips, and organizes. It moves laterally. And those are the things that happen because with the help of these enzymes. These enzymes are different enzymes. This is a typical phospholipid. It's a glycerophospholipid. It's a derivative of glycerol. And that's why it's called a glycerophospholipid. It has a phosphorus head group here. This is the phosphorus. And the head group is here. And this is in the glycerol nomenclature. You have 1, 2, 3, the second position. And this is the first fatty acid. It's called SN1 fatty acid. This is usually saturated fatty acid. This is, it is unsaturated fatty acid in biological membranes. That's a rule, but there are some exceptions. And here, this is the head group. The interesting thing here is, this one is mostly arachidonic acid that is 20 carbon long, and that plays a major role in your inflammation. And this enzyme that acts on this 2 position is called phospholipase A2. The nomenclature is simple. In the 1 position, the enzyme acts to remove the saturated fatty acid. It's called phospholipase A1. And here, there is an enzyme that acts here, removing the entire phosphorus in the head group. It's called phospholipase C. This is where inositol phospholipid metabolism comes. All the IP3s, IPs, and how the calcium regulations and all those things come here. And this another enzyme here only chops off the head group, and that's called phospholipase D. And these are the phospholipid hydrolyzing enzymes. These are called phospholipid hydrolases. And these are very important in the membrane lipid dynamics as well as cellular signaling and cellular function. As I mentioned earlier, unless phospholipase A2 acts on arachidonic acid and releases arachidonic acid, you don't have inflammatory reactions. Once the arachidonic acid is released, that arachidonic acid is metabolized by different sets of lipid oxygenases. And these lipid oxygenases, one of them is called cyclooxygenase, called COX, and the other one is called lipoxygenase, called LOX. These are the acronyms. And the cyclooxygenases make a number of compounds called prostaglandins. And the lipoxygenases make hydroperoxy fatty acid of arachidonic acid and converts them into leukotrienes. And these are not only inflammatory mediators, they're also autocoids. They have autocrine function, they have paracrine function. Still, we don't know what concentrations are required to drive an autocrine function, a paracrine function, and an inflammatory function. So these are very important. Don't forget one thing, inflammation is an adaptive reaction. You have to have inflammation. Inflammation is necessary to fight infection or to fight a disease condition. If it is overwhelmed, then it's a problem. So what happens is when you take ibuprofen, or when you take aspirin, or when you take indomethacin, or Tylenol, acetaminophen, what it does is it inhibits cyclooxygenase. That's how your pain is inhibited. See, these lipoxygenases are also very important in case of inflammation. Some of these asthma drugs, they are given to inhibit lipoxygenase. And these COXes are different ones, COX-1, COX-2, and also COX-3. Lipoxygenases are 3 different types, lipoxygenase 5, lipoxygenase 12, lipoxygenase 15, depending upon where they actually act on the substrate. So they generate several products. The reason why I'm showing it, because it has a bearing for my data I'll show you later. And these are actually the inflammatory mediators. These are very important in cellular function as well as cellular reactions to stress. One of the things is the vasculature. In the blood vessel, the innermost lining of the cells, the endothelium, is very important. I'll talk about the functions very soon. And this, what we observed earlier, because I was more focusing on reactive oxygen species and oxidative stress. At that time, I was interested whether oxidants cause any barrier dysfunction. And we found out that yes, they cause barrier dysfunction through the activation of the membrane phospholipases. So we know that under a scenario of oxidative stress, oxidants do cause barrier dysfunction through activation of membrane phospholipases like phospholipase A2 called PLA2, phospholipase C called PLC, phospholipase D called PLD. And these enzymes actually regulate the barrier function. If they're activated over a certain state, they actually cause endothelial barrier dysfunction. I'll tell you what is this barrier dysfunction is. Going back to one of the phospholipases, it's called phospholipase D or PLD. There are 2 isoforms present in mammalian systems. One is called PLD1, the other one is called PLD2. We still don't know their localizations. We still don't know much about it. It's a backwatered enzyme. We can't even isolate it. We can't purify it. We don't have any inhibitors for this enzyme. Very few laboratories work on this. We actually go with the kinetics, product formation. We have now antibodies. We can detect them. And we have genetically engineered enzyme. We can go and look at some of these functions. So typically a mammalian cell, including the humans, we have 2 different isoforms, PLD1 and PLD2. PLD1 is also present in the nucleus. PLD2 is present in the cytosol. They go to the membrane, they are translocated, and they act on the phospholipid to hydrolyze the head group. And this is a very powerful signaling enzyme. So what does it do? PLD acts on phosphatidylcholine. It's one of the phospholipids, makes a very potent bioactive lipid. These signal lipids are called bioactive lipids. And that one is called phosphatidic acid, PA. And then that PA is further metabolized to diacylglycerol or lysophosphatidic acid. These 2, LPA and DAG, are also very strong bioactive signal molecules. They're at nanomolar level in the body, even much lesser, and they attack the targets. What are the target proteins? Cytoskeleton. Now going back to the membrane, membrane is nothing but a phospholipid bilayer with integrated proteins and with receptors, with transporters, and cholesterol and other things. But one of the important things is the cytoskeleton. The cell has its own skeleton. The skeleton regulates the cell movements, cell shape. And this cytoskeleton is nothing but proteins. It has actin, just like muscular type of actin. It has myosin. It has a number of cytoskeletal proteins. These cytoskeletal proteins are actually targets for these lipid molecules. And when these are generated, the cell shape changes and the cell reorganizes. For example, in endothelial cell, the cytoskeleton is always required in action because the endothelial cell rolls, moves, angiogenesis occurs, new blood vessels form, because for angiogenesis, endothelial cell is the precursor. So how does it happen? The cytoskeleton is regulated, is regulating the angiogenesis. So the molecule, the signal molecule looks like this. This is phosphatidic acid. It's nothing but diacylglycerol phosphate. It still has, it only loses the head group, but still has the fatty acids in it. And the interesting thing is when the enzyme PLD acts on the phospholipid, forming phosphatidic acid, if you have a primary alcohol, either ethanol or 1-butanol or 1-propanol, it actually converts that to phosphatidyl alcohol. This is what they discovered in alcoholic patients. When in alcoholic patients, the blood levels of phosphatidylethanol is very high. They were wondering, where is this coming from? It's coming from PLD acting on phospholipid and making it phosphatidylethanol. So we exploit this as a tool to measure the enzyme activity. We add a little bit of alcohol, very small amount of alcohol, and then chase the phosphatidylethanol formation. And that's the right amount of phosphatidic acid formed because of phospholipase D action. Now, enough said about the phospholipase D, because I wanted to show you that there is an enzyme called phospholipase D. It's a powerful enzyme and at the membrane level. It forms phosphatidic acid, PA. The PA is a signal lipid. It's very important. Just I want to drive this. And now coming to the biological periodic table, there's so many 108, 110 elements present in the biological periodic table. When you look at these transition metals, every metal has a biochemical function. And some of the metals actually don't have a biochemical function. Some of the metals are toxic. And even the metals that are biochemically important, nutritionally important, they're also toxic. One thing is the coordination of these metal ions. Even the nutritionally important metals are also toxic. For a long time, I worked with a zinc expert, and my mentor, my old man, used to say, zinc is the least toxic metal. And I used to argue with him. How do you say that? But now it is increasingly becoming evident that zinc also causes some of the adverse reactions in the body. So it's very difficult to say whether transition metals or, the system, biological system has evolved to maintain a very delicate balance. We all have evolved from the sea, and what all the elements present in the sea, we have them. We can't have too much iodine. We can't have too little iodine. So that's how we have evolved to maintain a very delicate balance, and let's not disturb the balance. So that's what it is. Those transition metals are so important in that, and also both on the nutritional biochemical side as well as on the toxicology side. Again, there is a very thin line of separation between what is nutritionally important and what is toxicologically important. There is a very, very fine line where you can draw. This is one thing I always, show the examples of kids, because this is what happens with either the developed world or developing world, and both are same. And this is probably from India, where I see the lead batteries are collected in Calcutta somewhere. These lead batteries, kids are employed to recycle lead from the lead batteries. Nothing is given to them. And that's what this is, a metal of the brain. The reason is we all struggle. As a student of ecology, I think I learned one thing. 99% of the energy is allocated in the reproductive effort. That's the statement made by MacArthur at Princeton. 99% of the energy, yours, mine, even bacteria, is allocated in reproductive effort. Parents are sitting here, I'm a parent, who will be wondering, worrying, and my son is 25 years old, he's well off, but still we worry. And after we pass, everything goes to them. So that's actually, even in the cultured societies, that's reproductive effort. The idea here I'm driving is that we are transient. We are only storing the gene and passing it on. So that's what happens here is not good for the gene. That's, I just end my statement there. So now comes to the levels. The levels of lead. These are acceptable. This bothers me. What's an acceptable level? Acceptable level of consumption of alcohol, acceptable level of smoking marijuana. There is no acceptable. There is no limit. There is no, safe level. Yesterday I was talking to somebody. There is no safe level. What is in radiation? We learned one thing. What is the safe dose of radiation exposure? There is no safe dose of radiation exposure. Same way, there is no acceptable level. There is no acceptable level of carbohydrate consumption. So that's what it is. These these keep changing. That's what these legislators do. I'm sorry to say that that's what the governments do. That's what the organizations do. And they come up with these acceptable levels. I think these have to be revisited and they have to be strict. In a very advanced country like ours, if we come up with this, imagine what I showed previously in other countries where they're developing. And this is one of these things, the acceptable levels. This is the same case with mercury. Acceptable mercury levels in the environment. So that's a problem. Again, here lead. Again, Newsweek has a child. Again, I brought another child's picture here. So lead, what it does. Lead makes the mind give away. It's a Greek saying. Now coming to mercury, mercury gets converted to organic form. Organic form is much more toxic. Yeah, well, you know that. And that's what it is. Still we know, even in my culture back home, I was telling somebody that red oxide of mercury was given in Ayurvedic medicine. And we knew the problems there. Malformed babies. This again, another child picture. This is the Minamata disease. It's very well known. Just the fetal effect. It's a very powerful teratogen. Just showing some of those. So coming back to all these heavy metals, what they do, and you look at the membrane as the gateway of the cells, and there is oxidative stress when the heavy metals hit, especially I give you an example here, mercury, and then oxygen reactivity. The unsaturation is very highly reactive with the reactive oxygen species, and the unsaturated fatty acids are oxidized. You have lipid peroxidation, and then you have also generation of bioactive lipids through the activation of phospholipases, and then they cause membrane alterations and signaling. Each stage has a signaling step. These all will happen very quickly. You don't wait until 10 months of mercury exposure. It happens right when the person is exposed to this and with the minimal levels. That's what I was going to address here. Now coming to the cardiovascular system, you have both venous as well as the arterial system. You have these blood supplies. The very important one is the heart, the other one is brain, kidney, and the most important one is vascular plumbing work. Heart is a very, very tough tissue. When I remember heart mitochondria, I require about 5 millimolar hydrogen peroxide to kill it. Even isolated hearts don't stop beating. They're very tough. Heart muscle, that's the reason why heart muscle is there to do the job in our bodies. But the problem is the plumbing. The problem is the vasculature. That's why they're called cardiovascular diseases. Actually, they should be called vascular cardiac diseases. So what happens in the vasculature, if any damage occurs in the vasculature, and that reflects in the cardiovascular as well as cerebrovascular disorders. This is how it looks like. It's an artery, and it's a vein, venule, and you can see the endothelium, one monolayer, one cell layer thick, that's it. A monolayer of endothelium inside the lumen of the blood vessel. That's a very potent, and we call it endothelium as an organ. Even in the blood vessel, endothelium itself is an organ. And that's what it does. It functions. Actually, it maintains several functions, and it does maintain the structure of the blood vessel, the function of the blood vessel. It regulates the blood vessel growth by angiogenesis, because unless endothelial cells divide, you don't have new blood vessel formation. The capillaries, the small capillaries in the skin, the small capillaries in the lung, which is 4 tennis courts long. A lung, if you spread, it is 4 tennis courts long. That is the area of lung. It has nothing but profuse vasculature in it. All the gas exchanges, oxygen exchange, all the gas exchanges take place in the microcapillaries of the lung. And these microcapillaries are very, very important. One thing, those capillaries have no or little smooth muscle. They are all endothelium, the endothelial tubes. See how important is endothelium there. And they also help in clotting, blood clotting. And they are very helpful in inflammation and infection, leukocyte rolling, and other things. And they also modulate the state of the blood vessel in oxidations and other things. Above all, they maintain the permeability. You have leaky vessels because you have endothelial barrier dysfunction. Endothelium has a primary property as the barrier of the blood vessel. It does not allow macromolecules to pass through the lumen into the interstitium, through the smooth muscle cells. It is very tight. If you grow them in dish, after 2 days, they attain a morphology, the monolayer attains a morphology called cobblestone morphology, just like the bricks. And these are very tight, and the cytoskeleton plays a major role. If there is an attack of oxygen radicals or stress or heavy metals or toxins, the tightness, they form a gap. Through the gap, the macromolecules from the blood go through, the neutrophils go through, the macrophages go through. That's called diapedesis. It's called extravasation. The extravasation of macrophages from the blood in through the endothelial gaps into the interstitium causes inflammatory reactions. So that important is endothelium. Endothelium, one more function is it makes nitric oxide. Nitric oxide is a very potent vasodilator. If endothelial dysfunction occurs in smokers, there is endothelial dysfunction because they smoke and nitric oxide production is lowered. Smooth muscle cell doesn't get nitric oxide from the endothelium, so therefore there is no vasodilation. So endothelium is that important. This simple single monolayer thick of cells in the lumen inside the blood vessel regulates the function of the blood vessel. If that endothelium is damaged, you have problems. Vascular problems that lead to cardiac problems, that lead to cerebral problems. That's what happens. Atherosclerotic plaque actually has a tight interaction on the endothelial monolayer. That's what happens. So that much important it is. So now what are you going to say? You have talked about phospholipases, you have talked about heavy metals, talked about oxygen radicals, talked about endothelial barrier dysfunction. So where are you going now? So the current paradigm is, as Steve mentioned earlier, that cardiovascular diseases have been associated with heavy metals, especially mercury, for a long time. And we have been noticing this especially in the fish oils. And these fish oils contain heavy metals, especially mercury. The fish oil is collected from certain regions of the world. And not only that, if you have heavy metals, the omega fatty acids are not taken up by the cell properly. There is an epidemiological study done, fish oil consuming populations in Norway, because fish oils have high mercury levels, their omega fatty acid levels are low. Something it does for the omega fatty acids, there is 20 to 6, 20 to 5 fatty acids, these fish oils, their incorporation to the cell. This is actually inhibited by mercury. We are addressing that. We see some data, but I don't want to present here, but something is happening at the acylation. We call it acylation of the phospholipid. Something is happening, it's probably inhibiting one of those enzymes. So based on that, even occupationally, there are some reports that mercury workers have cardiovascular diseases, and also they have shown vascular abnormalities. They're published. I went back to this and thought, okay, fine, let's look at least some of the model systems, what mercury does to the vascular endothelial cells and vascular smooth muscle cells. The paradigm, the hypothesis was heavy metals like mercury activate phospholipases at the membrane, and these phospholipases generate bioactive lipid metabolites, and these bioactive lipid metabolites alter the endothelial function. So what comes first? Which phospholipase comes first? Which one is actually reactive first, or both of them go together? We don't know. Nobody has even shown that. Even in the normal setting, just recently, about 4 months ago, we published a paper that phospholipase A2 and phospholipase D and phospholipase C speak together. The lipid enzymes talk together. So maybe this is the same scenario. Well, let us look at it. That's the end. In the meantime, during the activation of these phospholipases by mercury, probably cyclooxygenases and lipoxygenases are involved, because mercury also causes inflammatory disorders. It actually activates the inflammatory cascades. So let's not ignore it, and let us look at those cyclooxygenase and lipoxygenase activations. So what typically we do is we take human endothelial cells or bovine endothelial cells. These are primary cells. These are not transformed cells. We grow them as monolayers and expose them to different types of mercury. In this case, we studied inorganic mercury called mercury chloride, organic mercury called methyl mercury, and another notorious candidate here, now people have opened eyes, thimerosal. And now autism is linked. There's a lot of debate, agreeing, not agreeing, disagreeing. But even then, for me, I wanted to look at thimerosal because it's a very potent mercury compound, organic compound, so let us look at that. Not only that, it's also a lot of preparations, pharmaceutical preparations have thimerosal. I used to inhale a nasal drop, nasal mist when I was 12, 13 years old, made in England, which had high levels of thimerosal. It's so bitter too. It goes through and I used to taste it. And I remember it has thimerosal. I used to read those ingredients. What is thimerosal? It's actually antibacterial, bacteriostatic agent. So you can see the activation. This is phenomenal activation. If you look at the controls, even the doses, like even 5, 10, 25 micromolar, you can even do it 1 micromolar. And if you take 10 micromolar and look at these different concentrations of different time points, you can look at 15 minutes. We don't even see with the tumor promoter. For Balester, we use it as a model to activate phospholipase D. It only activates up to here, 5,000. And with this, it's phenomenally high. That means it does so much devastating effect on the phospholipase D that it activates very momentarily, like even 5 minutes. So here I show 15 minutes, but I can show in 5 minutes it activates in the bovine endothelial cells. So what happens is now it activates. So what can we do? Is it a metal interaction? Or is it something else? So typically for a metal toxicologist, you throw some chelating agents and you see whether you can inhibit. This is pharmacologically important too. The reason I used chelating agents, I had the pharmacological standpoint of view in the back of my mind, and I said, okay, which chelating agent will stop? So I used EDTA. EDTA was not that effective to inhibit either mercury chloride one, it inhibited significantly, and a little bit on the thimerosal one, but methyl mercury, EDTA did not do anything. And then we also used PDTC, pyrrolidine dithiocarbamate. These are carbamic acid derivative. It's a strong metal chelating agent. And also it's an antioxidant. And we tried that. Methyl mercury was inhibited. All of them were significantly inhibited, but methyl mercury was inhibited much more. Sorry, mercury chloride was inhibited more. And we used another chelating agent called DETC. I was mentioning last night this one. And this one actually inhibited very significantly in all the 3 cases. So when you choose a chelating agent, you have to be careful what form of mercury it is present in the body. Always they measure mercury levels. This is the problem. They don't look at the speciation of mercury in the body. The speciation of mercury is very important. Is it still organic? Is it bound to organic acids in the body or in the blood? Is it bound to the proteins? What are the complexations? These are very important for a pharmacological standpoint of view. Because once it is bound there, you don't know what are metal ligand interactions. So speciation is very important. I think this was talked about 10 years ago in a toxicology meeting. Body speciation of mercury or any other transition metal is very important. Don't think that it is free. So that's what the chelating agents tell us. So next what we did was, what happens when you curb with thiol protectants? When you use a thiol protectant, what happens? Because mercury is known to attack sulfidyl groups, because it forms a very tight complexation with SH, protein SH as well as soluble SH like glutathione. So no matter what, whichever sulfidyl group, this is N-acetylcysteine. When you use N-acetylcysteine, almost completely eliminates the activation of phospholipase D. If you use the other one, dithiotretol, it's called DTT. It's a laboratory sulfidyl agent. We protect the sulfidyl groups on the proteins. You throw some, the proteins will retain their sulfidyl groups. DTT is also, we want to test whether another one does it. And this one on your left-hand side called bottom, this is another one, DMSA, dimercaptosuccinic acid. This is also a heavy metal sulfhydryl chelating protectant. And DMSA is given for heavy metal toxicity. These mercapto acids are given, and again, completely not the heavy metal mercury-induced phospholipase deactivation in endothelial cells. And the last one is the classic one, penicillamine. D-penicillamine is also a sulfhydryl containing heavy metal protective agent that also protected it. This is where the N-acetylcysteine as well as glutathione protection comes into picture. The liposomal glutathione, the reason why it actually protects is because probably it's also curbing the reactions that happen for the heavy metals at the membrane levels, as well as quenching the mercury. Okay, this is what quenching the heavy metal. So that's the way to go probably, and also I'm pretty excited looking at how you target both lipids as well as the heavy metals in the cellular target level. So okay, chelating agents protect, sulfhydryl agents protect. So what about antioxidants? So when we tried different antioxidants, vitamin C, again, vitamin C only protected up to some extent, almost protected mercury chloride-induced activation of PLD, whereas in methyl mercury, it didn't do anything. When you look at thimerosal, again, significantly protected, vitamin C. So vitamin C across is not a good antioxidant, because the problem with vitamin C, I call it is a redox active antioxidant. That means it's a pro-oxidant. It's not only an antioxidant, it also forms oxidants. So you have to be careful when you utilize vitamin C as an antioxidant. And when you take the other one, a natural product called propyl gallate, it's a gallic acid derivative. It's very similar to the T polyphenols. It's a phenol, like epigallocatechin gallate. I don't know how many of you have known that. The epigallocatechin gallate family, this member this is called propyl gallate. Propyl gallate now completely eliminated mercury chloride, at least significantly in methyl mercury and not in thimerosal that much. Remember one thing, certain antioxidants like polyphenols are powerful antioxidants, and they're also metal chelating agents. They complex with metals, very tight complexes they form. Catechin, rutin, quercetin, gallic acid derivatives. These all form tight complexes with the heavy transition metals. So they do multifunctional protection. They offer protection in different ways. They're antioxidants because they are phenolic in nature, and they have hydroxyl groups that they bind with the metal ions. So the people have to look into those things as heavy metal protective agents, these natural products. That's why diet is very important. When a person has heavy metal toxicity or overload of heavy metals, the diet is also important. What type of diets they have to eat, green vegetables or more lycopene or whatever it is. So we have to think about those things. And the last one is a test. What we do is extracellular hydrogen peroxide, whether it is generated, we throw some catalase, bovine catalase, and see a catalase protected. That told us, okay, probably mercury in all three forms or in one form is causing reactive oxygen species formation in endothelial cells. So when we looked at that, in a dose response in an hour, you can see an enormous amount of reactive oxygen species formation in these cells. And if you treat with N-acetylcysteine, it's almost totally inhibited. The reactive oxygen species formation is inhibited by methyl mercury induction. And here, when you look at this, there is a superoxide dismutase mimetic called manganese T-BOP, and that goes inside the cells and behaves like superoxide dismutase. That also protected. So this confirms that mercury, at least methyl mercury, causes here reactive oxygen species formation in endothelial cells. So now, going back to mercury, why NAC offers such a tremendous protection? Why dimercaptosuccinic acid, DPP? Because we found out that the sulfhydryl compounds are protecting endothelial cell phospholipase deactivation under mercury insult. So what happens to the thiols? So when we measured the thiols, all the thiols are significantly down in these endothelial cells when treated with mercury compound. So the target, the primary target here looks like the thiols are actually quenched and then the enzyme is activated. So the conclusion of this part of this talk is mercury actually causes sulfhydryl groups quenching, and the membrane protein thiols are quenched. There is a structural conformational change of signaling happening here because of reduction in the thiol levels at the membrane level. So phospholipase D is activated and phosphoric acid is formed that leads to cytotoxicity or vascular toxicity, and further down leads to the cardiovascular diseases. So what happens here, the other story is, there is a tight relationship between mercury and calcium. Calcium is a paradox. Calcium is required, calcium is not required. Calcium is required for biochemical functions. Calcium is a notorious candidate in certain pathophysiological events. So calcium, and there are instances that mercury does interfere with calcium metabolism in the body. We know that ryanodine receptors, it's a good story, ryanodine receptors have like 70% of sulfhydryl groups. And these ryanodine receptors are targets for mercury. So based on that, we wanted to look at whether calcium is also altered by mercury during activation of phospholipase D. So one thing is, calcium is extracellular, calcium is intracellular. The extracellular calcium is quenched by a chelating agent called EGTA. EGTA does not get inside the cell, it only quenches the calcium outside. Whereas BAPTA, this is an organic quencher, it quenches inside. So when you treat these cells and look at phospholipase D activation, BAPTA as well as EGTA, both of them quenched PLD activation, indicating that both intracellular as well as extracellular calcium is required for the activation of the enzyme under mercury stress. So that tells that mercury actually perturbs calcium and activates phospholipase D. And then when we remove calcium from the medium, in the calcium deficient medium, there is no activation even if you put mercury. But by the time you add calcium, the activation goes up. So calcium is very tightly associated with mercury-induced phospholipase D activation. And when we titrate back, add calcium from the deficient medium, again you see the activation. So the calcium channel blockers, these are voltage-gated channel blockers. When you block them, you can see the inhibition of phospholipase D activity. And different, again, this is another one, another channel blocker called diltiazem that is used in cardiovascular disorders, cardiac disorders. Again, that protects it. And the other one is nimodipine, also protects phospholipase D activation by mercury. So calcium channels are important. When you completely block the calmodulin, again, you can see the inhibition. So calmodulin is involved. So these are the calcium levels inside the cell. When you treat with mercury, the calcium goes up inside the cell. So that mercury alters the calcium, so elevates the calcium levels in the cell through the calcium-gated channel. So what happens is when the endothelial cell is hit by mercury, voltage-gated calcium channels are opened up and extracellular calcium gets inside, intracellular calcium elevation occurs, then that activates phospholipase D and causes the activation of the enzyme. So now the calcium story is also coming. So calcium is very tightly involved. It's actually rattled by mercury, and calcium levels increase that activates phospholipase D. And very quickly, in 5 minutes, I will talk about phospholipase A2. Previously, you have seen phospholipase D activation by mercury. Now phospholipase A2, which is important in the inflammation. So mercury, again here methylmercury activates phospholipase A2 and releases arachidonic acid. And this one is again protected by mercaptosuccinic acid as well as PDTC. These are metal chelating agents. And again, N-acetylcysteine completely inhibited phospholipase A2 mediated arachidonic acid release, as well as propyl gallate, an antioxidant, again inhibited that. So PLA2 activity is inhibited by sulfhydryl antioxidants. And you can also see the phospholipase A2 specific inhibitors showing that PLA2 is activated by mercury. They also significantly inhibit. That means mercury activates CPLA2, that is cytosolic PLA2. Again, sulfhydryl groups go down, thiols, total thiols go down by methylmercury. Again, by treating with NAC, you can actually elevate them back to the normal levels. this is what happens in cell morphology. We always look at cell morphology primarily because the cell morphology tells us something, what's happening in the cell. It tells us the cytoskeleton is altered, and you can see with the different concentrations beginning with 5 micromolar of methyl mercury, how the endothelial cells are altered. It induces lipid peroxidation. Again, in endothelial cells, you can see the lipid peroxides going up. Methyl mercury causes enhanced lipid peroxidation. And the cytotoxicity caused by methyl mercury is inhibited by phospholipase A2 inhibitors. That tells that phospholipase A2 is activated by methyl mercury that causes cytotoxicity through these events. So again, if you look at another specific PLA2 inhibitor, the cytotoxicity is down. You can see that completely inhibited, at least significantly inhibited here. So the PLA2 actually regulates the cytotoxicity under mercury in these endothelial cells. The prostaglandins, you can see the enormous levels of prostaglandins. These inflammatory mediators are formed by methyl mercury inside the endothelial cell. So these come from PLA2 through the cyclooxygenase pathway. These are prostaglandins. This is thromboxane A2. Both of them were notorious. And you can see 8-isoprostane. 8-isoprostane also goes up. This comes from, it's a lipid peroxidation marker, very similar to prostaglandins. And this also goes up in case of methyl mercury treatment in the endothelial cells. So the thing is, the phospholipase A2 is activated by methyl mercury. Phospholipase D is activated by methyl mercury. So what is the connection? And both are linked in cytotoxicity. So which one comes first? So we went and looked at phospholipase A2 probably regulates upstream of PLD activation in mercury-treated endothelial cells. Let me quickly show that. When we look at PLD activation and inhibit that with PLA2 inhibitor, you can completely inhibit that. So the PLD activation by mercury is inhibited by PLA2 inhibitor. So that shows that PLA2 is upstream of PLD. And when you use COX inhibitors, cyclooxygenase inhibitors, something happened in the computer transfer. And if you take the cyclooxygenase inhibitors, this is indomethacin, and this one is CDC, I think. Both of them are CDC, in this case, COX2 inhibitor, completely inhibited PLD activation, showing that cyclooxygenase is upstream of phospholipase D activation. So when you hit endothelial cell, PLA2 is activated, and then cyclooxygenase uses that arachidonic acid, makes prostaglandins, and these prostaglandins cause the cytotoxicity and activation of PLD to generate phosphoric acid. So if you look at lipoxygenase inhibitors, interestingly, lipoxygenase inhibitors completely at a higher dose completely inhibited the PLD activation by methyl mercury in endothelial cells. These are two different lipoxygenase inhibitors we used here. And again, another set of inhibitors we wanted to check which one is effective. Again, these are lipoxygenase inhibitors, inhibited PLD activation by methyl mercury in endothelial cells, showing that lipoxygenase is also upstream. So these are the products that we measured. Again, these are measured by LCMS, liquid chromatography mass spectrometry. We looked at lipoxygenase metabolites, look at the increase in these metabolites, then treated with methyl mercury in the endothelial cells. And this one is a 12-lipoxygenase product, again, slight increase with the 12-lipoxygenase product. So the bottom line here is that what is the essence of this story is that mercury activates phospholipase A2 upstream of phospholipase D, releases arachidonic acid. That arachidonic acid is metabolized into cyclooxygenase and lipoxygenase metabolites. And those lipoxygenase and cyclooxygenase metabolites trigger phosphoric acid formation through PLD. So this many enzymes, PLA2 to cyclooxygenase, lipoxygenase to phospholipase D. Once phospholipase D is activated, you have cytoskeletal problems. Once cytoskeleton is hit, because phosphoric acid, PA, is known to affect cytoskeleton. So PA that is released by phospholipase D, once it is released by phospholipase D, that PA, that phosphoric acid, it's a bioactive lipid, and that one targets cytoskeleton. And once the cytoskeleton is attacked, the actin stress fiber is formed. We have the data, I didn't bring it. I need another 1 more hour. And that cytoskeleton, actin cytoskeleton polymerizes, and actin pulls the cell inside, and the cell loses its morphology, and gaps are formed, and there's vascular leak. So that's what happens. So PLA2 is upstream of mercury-induced phospholipase activation in endothelium of vasculature, and then it triggers cyclooxygenase and lipoxygenase. They make eicosanoids as well as prostaglandins and leukotrienes, and then they activate phospholipase D, and the phospholipase D generates phosphoric acid. That phosphoric acid alters cytoskeleton. That cytoskeleton alteration leads to cell morphology change, and that cell morphology change will lead to gap formation in the endothelium, and that gap formation is the cause for vascular leak and other inflammatory events. That is the gist of the whole story. So now we'll show you some micrographs, what happens to the PLA2 inside the cells. You control no mercury, no methylmercury. When you treat 1 micromolar, 5 micromolar, 10 micromolar, you can see the translocation of the enzyme so evident in these cells. This is PLA2, normal phospholipase A2. The activation, we look at phosphorylation. We look at phosphorylation of PLA2 because PLA2 is phosphorylated by a kinase that we think it's a MAP kinase, and that MAP kinase phosphorylates PLA2 and activates. You can see so intense in case of higher concentrations. So this is another signaling going on. If somebody asks me a question, how the PLA2, how the PLD are activated by mercury, this is the way they are activated. That means there is something else ahead of these phospholipases. When methylmercury hits, there is a kinase that gets activated. That kinase phosphorylates these enzymes. These enzymes then get activated and do their job. So that's why I wanted to show the phosphorylation sites. We're very heavily working on it, and we're going to, I just wanted to show, some of the data is not published, but I just wanted to share this with you, what's going on. The next one, this one here is the enzyme. One, I told you earlier, PLD is in 2 isoforms, D1 and D2. Just 1 minute, please. And D1 and D2, and D1, when we looked at phosphorylation of D1, you can see phosphorylation of D1 is also increased. That tells us that this also gets activated through a kinase mechanism. So the bottom line is here again, the mercury alters calcium. Calcium again is involved in PLA2. Arachidonic acid is released. Arachidonic acid is metabolized into prostaglandins and leukotrienes, then leads to the activation of PLD and membrane damage, and then blood vessel leak. Okay, I need to thank a number of my students here, and all of these. Joe Hild is a medical student in Kentucky, and Vivek Kuppasamy, actually, he was as a high school student, he started working with me in the laboratory, and he was the one who started on mercury chloride work. He is now doing engineering at University of Maryland. Emily Steinauer, she was with me for 4 years, and she did all the barrier dysfunction work, and she's now in Emory. Bruce Kaufman is in Toledo doing MD, and he did some work. Elon Peltz did undergraduate thesis with me. He did the calcium story. Jessie is in Vanderbilt doing PhD. She did all the methylmercury story. Tom Hagel, who won the Denman Award for this topic, he is now in Wright State doing his MD. Valerie and Shriek and Tim Eubank did the angiogenesis work. Lynn Sawyers did all the cytoskeleton work. Thank you very much. Thanks a lot.